Okay, we are recording.
Yeah.
Hey!
No, it's so I'm like should be out of quarantine, but I can't attend class because I haven't gotten an email from the health organization. Like I'm back in my room and all of this, but so bro, can you take notes for me again, or like send me pictures of the notes? Because I want to try and do the best I can on this side, but
So far, you're the only person on Teams today, so it might be better.
I got you.
thank you.
So last time, we had been talking about how flu virus mutates and about how flu virus can accumulate these random mutations in the spike proteins that let it still infect but not get recognized. So you can have a slight change that allows the flu virus to still attach and start the viral life cycle, but not be recognized by our immune system. So that kind of small mutation and that kind of small change is called antigenic drift. And antigenic drift slightly changes a spike shape. So it slightly changes a spike shape so that a virus can hide from the immune system. And this is why we have to keep having new flu vaccines every year, because flu virus mutates slightly over time. And somebody last time, maybe it was Sam, asked, well, how do they know how it's going to mutate? It's an educated guess. And so every year, usually in March, flu specialists from all over the world kind of get together and they have a meeting and they try to predict what next year's flu virus is likely to look like. And they come up with their recommendation and they develop the plans for kind of a mixed vaccine. So we'll usually have 3 different spike type of shapes. And because it's a slow process to make that vaccine, from April through July is the production time to get however many million doses of flu vaccine that you need for the whole world, basically, so that you can start giving it to people in August, September, October, like right now. Some years they guess well, and some years they don't guess well. So flu vaccine is not equally effective year to year, but it is partially effective always. And we'll talk more about vaccines toward the end of the semester.
I have a question.
Uh-huh.
So on the next slide, it says shift.
Yeah.
So
Shift,
yeah, it says shift.
something so different.
Okay.
They almost sound the same, but they're not. So antigenic drift is when you get the slight change in a spike where it's a little bit different. Same basic spike shape, but it's a little bit different. Antigenic shift is something else. This slide is describing antigenic shift. Antigenic shift is when you get a mix and match, a reassortment, a random grab bag of different spikes from different viruses. Antigenic shift can make a big change in a spike protein. And what this picture is trying to show is there's a virus that infects ducks, birds. There's a virus that infects people. The duck virus cannot infect people. So this is the original situation. Bird virus infects birds. People virus infects people. Sometimes you can have viral mixing in an intermediate host, or a host that could have both viruses infected at the same time. And for some reason, pigs are good at this, or bad at this, however you want to think about it. So this pig, this poor pig, it's infected with a bird virus and it's infected with a human virus. And in this pig, during synthesis, you get hybrid viruses. Hybrid viruses. And those hybrid viruses may have some pieces from bird virus, some pieces from human virus. But you could end up, once in a while, with a virus that can infect, or with a human version that's got a duck spike. And it's like it's never before seen in humans. This has always just been, kind of theoretical most years. This is what happened with coronavirus. This is what happened with coronavirus, except it wasn't a duck. I'm not going to be able to draw a bat on this screen. It was a bat, most likely. And what was the intermediate thing? You read this? It's unknown, actually.
Oh. Trick question.
How dorked out about this you are. It might be an animal called a civet, which is an Asian mammal, C-I-V-E-T, civet. But we know that the coronavirus basically came out of bats. It's not lab generated. Not lab generated. Came out of bats. There was probably an in-between organism, and now it infects people, and no people, well, back in the winter, no people on Earth had ever experienced that kind of virus before. So that's making it more deadly overall. We don't have immune experience with this in human history. This is called antigenic shift, and antigenic shift is the major way that viruses jump species. Or viruses suddenly appear in new hosts where they were not in that host before. This is much less common than antigenic drift. Antigenic drift happens all the time, but this changes the whole entire host for what a virus can actually infect. Antigenic shift. And antigenic shift requires that you have this intermediate host organism that can be co-infected, two viruses at once, and it can produce these hybrid viruses. Most of the hybrid viruses that would come out of such a situation are going to be crappy and not infective anyway. But it just takes one. It's like, oh, I've got a whole new host available to me, people. That becomes a big deal. Had some other thought in my mind, but now I don't know what it was. Antigenic shift. Oh, I know what the thought was. And this I'll have to fit over here. Antigenic shift is more likely when humans and multiple animals are living in high density together. This is one reason why it seems like viruses tend to come out of Chinese wet market areas. So this, what I'm calling wet market, it's kind of like their version of a grocery store. Kind of like a farmer's market. It's kind of like a farmer's market except with animals. So you can go buy your civets over here, and you can buy your fish over here, and you can buy your pigs here. And it's just a lot of animal material together in high density with a lot of people. And that makes this, this type of event more likely. So one of the major goals of public health virology is to not have those kind of wet markets and to try to discourage people from eating exotic game, let's say, like bats or civets or pangolins. Pangolins is the other animal that might be the intermediate host. What were you gonna say?
What is a civet?
A civet looks like a weasel. That is the best I can tell you. Professor Rudd would know so much more about that. It is a mammal. I think it is in the weasel family.
I was just curious because I had never heard of a civet.
Yeah, it's not, they don't live here, so. Yes. So that's pretty important to how coronavirus got started. Keep in mind that the rest of these prion-based slides, we're not talking about that this year. I'm going to switch to slides specifically about coronavirus. Yeah, go ahead and ask.
It's about,
So is this the SARS-CoV-2 slides or the influenza one slides?
I'm moving to the SARS-CoV-2 update slides.
Sweet, thank you.
And it's a really big file, so it's probably going to take a minute to load up.
but my question before I was really interrupted was, so the, I don't know how to pronounce it, I'm from Chicago, so the light cycle and the,
Lytic and lysogenic.
yes, the only difference is between the penetration, how the lysogenic, did I say it right?
Uh-huh.
Cycle has its DNA interpreted into the other genome, so it's a long-term effect, but it still goes through everything else.
Having the genome injected as opposed to the whole virus is not specific to the lysogenic cycle.
Okay,
That's just the example that they were using. It went that way.
okay.
So before I announcement, oh, I know where that comes from. Before I talk more about coronavirus, lytic cycle and lysogenic cycle for viruses, they both would do adsorption or attachment. They both would do penetration step, and that could be different styles depending on different virus. But then at that point, lysogenic are able to integrate into the host genome and stay there or wait there and go into a latent state. And then at any unpredictable point, which we will just call sometime they could actually go back and rejoin the rest of the steps. And I don't think I made that clear in when I was talking about this. So.
Because I was just,
Brooke first and then Hawkins.
I was just wondering because on the picture, it just feels like it's just going through the whole thing, but then at the bottom, it just only shows like the difference in the penetration. So I was just wondering.
Yeah, this arrow that viruses in lysogenic state can actually do that. I don't think I said it, but it's really important. So if you think about this related to, let's say HIV, people can get infected with HIV and not know it, but it's already integrated in their genome. And then some number of years later,
It can just.
Then they start to feel the effects because the virus has re-entered lytic cycle.
Okay. So it
Yeah.
re-enters into lytic cycle?
Yeah,
Okay,
yeah.
okay.
If it always just kind of stayed in lysogenic, it wouldn't be as bad. But they eventually
Go
will
back.
reactivate
Okay.
and go through lytic cycle. But most viruses in the world can only do lytic cycle. There's a few that can go into lysogenic cycle, wait for some period. They're altering our genome. At some point, they can activate and start making virus again. And that can, you can go in and out too. Like HPV works that way.
Okay.
In and out of lytic and lysogenic. Yeah, Hawken, what was your question?
Oh, you kind of answered. I was just gonna be like, so the viruses like they wait until like the opportune moment, or like when the conditions are best in the host. Like for like if there's a cold, like if a person has cold sores, like they're most likely to get them when they're stressed or sick.
There's something to do with being stressed and sick that makes the virus reactivate, but how it knows is still a research question.
Okay.
But we know that correlation is there for cold sores, like you say.
Mm-hmm.
For HIV, we don't know what makes it come out of the latent state and start going back into lytic state.
Okay.
I bet there's a lot of people researching that. So this announcement stuff does not have to do with you. I did this presentation in science seminar a couple weeks ago, and these people are having virtual interviews today, plus Liz White.
Good for her.
Yeah. Okay, so the reason I developed this in the first place is Dr. Vaughn said, why don't you do a science seminar about SARS-CoV-2, because there's a lot of things that we might think that students know about it that they don't know. So this is going through a lot of basics of it, and I thought I would put this dumb meme picture as an example of misinformation. I love this, and this is a totally reasonable question too. If we can smell through masks, which we can, although I have watched my own husband in Target like go like this so he could sniff the candle better. You're gonna buy a fall candle. We can smell through masks, but they block COVID. How does that work? So that's something I'm gonna talk about in this, and just some other info about coronavirus. So first thing, the name of the virus, the actual virus name is SARS-CoV-2. It stands for Severe Acquired Respiratory Syndrome. CoV is for coronavirus. Yeah, I'll try to write it up here. Severe acquired respiratory syndrome. That's the SARS part. CoV is coronavirus. And 2 is because this is not the first one ever discovered, surprisingly. There was another one several years ago that infected some people, but only maybe like 10,000 people, and then it was gotten under control and the world moved on. This is the second one. This one is not under control yet. So that's the name of the virus. And coronavirus is a whole family of viruses, and they're named for corona crown, like corona meaning crown. And the crown is referencing the appearance of the spikes all over the virus. Kind of like a 3D pretty crown going around. So the virus is SARS-CoV-2. This virus infects people through their respiratory tract. Through their respiratory tract, and this is not to scale. That's ridiculous, but I wanted you to be able to see it. And I actually made these slides first for 5th graders. So once in a while, if you feel like, they're originally made for 5th graders. So you can breathe it in through your nose. You can breathe it in through your mouth. It's possible that we can contract it through our eyes. So it gets on the surface of your eye. You know, you have that drainage down into your nose. It's possible it can infect that way. That's why we're having you wear safety goggles. That's great. And your safety glasses in the lab. But it's a respiratory virus. Go ahead and breathe it out. Go ahead and write this down. You can do not breathe it out. You can breathe it out through your mouth. Let me redraw that. Oh, what happened? I'm holding the eraser. Okay, you can breathe it out through your mouth. You can breathe it out through your nose. And I really appreciate you all in this class. You don't ever have your nose hanging over your mask. That drives me crazy. That drives me crazy. You need your mask over both parts because we breathe it out through both parts. So this virus, we breathe it in. It can attach to certain receptors on the surface of our cells called ACE receptors. So I'll just, there we go. Throw that there. ACE2 receptors are what the spike binds to. And I think those of you that had AMP2 with me, Brooke and Constant, I did not talk enough about ACE2 receptors previously, but I'm going to now. So if that doesn't ring a bell, it's because I didn't teach it back then. But that's the receptor. And if the virus can bind to that receptor, it can get into the cells, and then it will cause an infection. Some of the infections can be silent. So if the infection is silent, we say that the person is asymptomatic. Asymptomatic. And unfortunately, or maybe this is fortunate, it's probably fortunate, SARS-CoV-2 infections, a lot of people that have it are asymptomatic. And you don't know that you have it, but you might have it. Some people never feel a thing. So some of the students I've had who tested positive, they never felt bad. They felt fine always. I've had other students that tested positive and they felt really pretty bad. All the ones so far that felt pretty bad, they are okay now, is good news. But if the infection causes symptoms, that's when we call it COVID-19. So COVID-19 is the name of the symptoms or the disease. SARS-CoV-2 is the name of the virus. You will hear people out in the public like using the term coronavirus and COVID like they're the same thing. And that's basically okay for them, but as medical people, you should go ahead and call COVID the disease. Coronavirus is the virus, but the real name of the coronavirus is SARS-CoV-2. So that kind of thing, that will be on the exam, that terminology. And I have to remember today that I need to give you all the exam review today, not wait too long, or the exam topics list. Okay, so as far as symptoms, you have these ACE2 receptors in different places in your body. You have them in the lungs and also kind of in your lower respiratory system generally. You have them in your heart, which is interesting. And you also have them in your blood vessels. Let's see if I can write that on here. Heart, lungs, blood vessels around your body. So if the virus infects cells in those regions and causes a lot of damage either directly or indirectly, these are the major symptoms. So notice here, pneumonia. This is from a Greek medical journal, and they apparently call it pneumonia. It's pneumonia. This is respiratory distress syndrome. So those are the breathing issues. This is low blood oxygen. Hypoxemia or hypoxia. Low blood oxygen. Myocardial injury, that's heart injury. And these are the major symptoms. One other possible major symptom is extreme low blood pressure, which is very dangerous. That comes from what ACE2 receptors' normal function is. It has to do with blood pressure. So the major symptoms that are the most concerning are breathing problems, heart damage, low blood oxygen. Make yourself a note here. Oh, goodness. Some of these symptoms seem like they are worse for people if the people's immune system are overreacting to this. And that will make more sense when we learn about immune system. But like Liv and Chelsea, all the inflammation responses, you can imagine if that got too severe in the lungs and it couldn't be controlled and you're having fluid in the lungs, the overreaction can be deadly. So we'll come back and talk about that when we get to the immune system. But it seems like the people who do best with SARS-CoV-2 infection, their immune system fights it off at just the right timing. And it doesn't go too far through the body, and it doesn't cause a huge damaging immune fight all over the body. Have any of you had people that you know or in your family or that have been really sick? Okay, it happens. This summer we had a faculty member at King get it, and they are just, this was like in July, and they are just now starting to get their energy back and be normal. It's not a teaching faculty that you all would ever be in contact with, but, and they're healthy. I mean, they were way healthier than me. Yeah.
So if a person has like a really bad allergy to something, so their body like overreacts to the allergies, are they more likely to overreact when they have that?
So far, no. So far, having allergies is not correlated with worse COVID-19 outcomes, is how I would say that. And the other interesting thing is that having asthma is not correlated with worse COVID outcomes, which is surprising. I had that question in seminar, and I know a lot of people have asthma, but for some reason, asthma does not make COVID outcomes worse. I've had that question like multiple times this summer with people like, my child has asthma, should they go back to school? Oh my gosh, I don't want to make that decision for you. But that's what I say is, so far, asthma is not correlated with worse outcomes. Sam, did you have a question? Okay. Oh, I saw your hand move. Okay, this is from the 5th grade slides. This is talking about being asymptomatic. So people that have the virus and still feel healthy are asymptomatic, and we call them carriers. And the reason, if you've had to be quarantined for so many days, it's because just to make sure that you're not a carrier, let some time pass, make sure that you're not going to get it, and then you can rejoin everybody else and not pass it to them. So I told during seminar, all of you that have been in quarantine, or all of you who have stayed home a few days, I'm proud of you, and that's very difficult, but it's really important, and it's working.
So how did they come up with 14 days as the time period that you need to stay in quarantine?
That comes from observing people starting back in February in different countries who tested positive and doing contact tracing and figuring out, okay, if they were positive and this is all the people that they were around, and looking for when other people could catch it and looking at that in big populations.
Okay.
That sounds kind of complicated, and it is, but it's from doing really careful contact tracing and careful record keeping. And that's one of the things that has not really changed this whole time, is the 14-day thing.
Yeah.
It's pretty reliable. It's not, that doesn't mean that you can't transmit it on day 16.
I was gonna say, so, well, it can't just go away after 14 days, right? Or is it?
You're
like-
not shedding it as much. So let me go back a few pictures.
That's kind of what I was gonna ask, is
Yeah.
like, is there a point where it's kind of worked through your system and you're no longer contagious, but you're still like working it out? Like other
Yeah,
things?
so this 14 days thing,
Mhm.
it's way easier to look in populations and see who's spreading it to who than it is to actually go in and monitor people individually and say, how many days can we still detect it in your body?
Yeah.
The detecting in the body lasts a lot longer than the actual spreading to other people. So you don't have to write this down, but the coming back out, so based on what we know so far, the transmission is most about 2 days before symptoms is when it seems to be the most,
So that's
or the
why
highest, and then that continues up until maybe 6 days after you show symptoms.
So that's why we contact trace for the past 48
Yeah.
hours?
Yeah. Yeah.
Okay.
Yeah, all those numbers of how long to quarantine and how far back to contact trace, it's coming from really careful record keeping of people who've had it. Mm-hmm.
So, since you can breathe it out and people can catch it from you just breathing, why is the test they have to stick it so far up the nose? Could they not just swallow them out?
So I didn't know this until last week. How many of you have had the eye stinging deep test? I mean, like when they put it so far back and your eyes water. Okay. How many of you had the shallow test? Okay, so the ones that King is doing in-house right now only has to go 1 inch into your nose. And so some of us in biology had heard students say, I got the deep test, mine only went that far. And we're like, oh my gosh, like, is this a problem? So I checked with our we have this nursing faculty that's just amazing, and she's over the COVID response. So I checked with her, and she said that there's different tests, and they go at different depths. But you want to be able to detect it 95% of the time. So the first tests that were developed, you had to get all the way back here.
To the brain.
That's the one that Moses had, and I didn't tell him that they were going to do that because he worries about stuff.
Oh.
I didn't tell him, and then the doctor was just so great. She was just kind of talk, and she
Oh, no.
like, did it really quick, and then his eyes were watering. But I found out later he thought it was going to be a blood test. And he was worried about that, so I should have told him. But now there's better tests developed, and it only has to go outside.
I saw a video of one guy, he got the test in his nose, but then like, he breathed in and he got
You ought to have a sneeze out reflex. When
Yeah,
that
no,
does
I
that.
Well,
But that's why, so better tests are being developed every month.
Okay, honestly,
Yeah.
I got tested like back in June, and they did like both of my nostrils, like
Both?
deep. Yeah. So like some people do like one, but then other tests do like both.
It depends on the test. Like each individual test has been developed and tested to see how reliable it is, and they have different instructions. Mm-hmm.
Okay, so, when I was in Chicago, right, I had to get tested like 3 times, okay, and every single time was different. Like I had to do it myself and like touch my brain and each nostril, and then like I had to like swab my mouth. So like I had COVID like before I came here,
Mm-hmm.
but my test still ended up being positive. Like after my 2-week, like
Yeah.
my 2-week length,
Yeah,
I got tested again and got tested positive,
yeah.
and then quarantined again.
Oh, because
then-
you tested positive?
Yeah, for a second time.
Yeah, I'm
And then
sorry
third time I
about
got
that.
Positive.
It's totally possible to
Yeah,
be.
I've been 3 times.
Yeah, so, sorry. There's a difference between what the tests are testing for, and I saw your hand up, and I'll get your question next. So, you can test for viral genome, which is RNA in this case. So when you get swabbed, that's what they're testing for. That is not the same thing as a test for infectious virus coming out of you. These are fairly easy to do. This is very difficult to do. Very difficult to do. And so this is going to happen to a lot of people, that they would still technically test positive because they've still got virus RNA in their body.
Mhm.
It's not the same as being infectious.
Okay.
And when you go between different states with different rules, where this is a new disease, some of that might be different rules in different states. The other kind of test is you can test for blood antibodies, which means that the people have had an immune response in the past. Oops.
And how long do those antibodies stay in your system?
Let's see, this would be relevant from about 2 weeks after infection. Let's say 2 weeks until, we don't know. We don't know yet.
There's no, like, lifespan of the antibodies. I heard like.
There is, we just don't know what it is yet, reliably.
Okay. Because like my sister, she had to get tested before she
Mhm.
went to school, and she came back and she had the antibodies on her
Yeah.
test.
Yeah,
And
which means she was
And she was
exposed.
sick in like November.
Yeah,
So
yeah, it's seeming
I
like
just wasn't sure like if that was,
the virus was here before you realized.
Mhm.
You said November?
yeah.
Yeah, or she could have had it since then and just never
Yeah.
felt it as well.
I'll just say, because she went in November and they told her that she had like pneumonia.
Mm-hmm. I mean, she had pneumonia. It could have been from COVID or not. So this was from back around March, that a big hospital in
dang.
New York. Oh.
Oh, man, I don't know, my birthday.
You didn't have a question? Okay, I'll let you. So a big, rich hospital in New York that has a lot of resources decided to test every woman coming in to give birth, like in a 2-week period. And this is, generally healthy people. Everybody that was going to give birth. 15% of them actually were infected. And I remember reading this in the news, and everybody was like, "Oh no, this is terrible." 15% of the people were actually infected. Most of them didn't feel a thing. A few had some mild symptoms. So this was kind of a snapshot of how many people in New York Metro were infected at that time, using this as a snapshot. So there are a lot more people infected than test positive or who have symptoms. And so if you find, like last week at King, we had 25 positive. There's a lot more people that were infected, but that's how many people were detected. But also, the spread is social stuff at night. It's not classroom. I'm pretty confident in that, so I'm not worried about getting it from you all in the classroom. Because we're wearing masks. So.
So when they do the test, like now they have the rapid test and then they
Mhm.
still have the test that you send off
Oh,
and
yeah.
it takes like
Months.
Yeah.
3 days, whatever. Like, which one is more accurate? Is the rapid test accurate? Is
Okay,
it?
so rapid tests are detecting actual spike proteins. And so those are the ones where maybe it takes 30 minutes or 15 minutes, and apparently the Virginia side, people can get those all the time.
Yeah. I was gonna say a lot of urgent cares.
No, too bad, Tennessee.
What are you talking about?
If you live on the Virginia side, you can go to the health department and get a rapid test anytime. Unfortunately, King's in Tennessee. So,
I was gonna say,
no
they
rapid
have
tests
those.
for us.
They have those at the urgent cares at home.
Yeah, it depends on the state.
Yeah.
The patchiness of what states are doing is kind of a drawback. It's different everywhere. These are less reliable, but they're pretty good. These are more reliable, but they're slower. This is slower, but it's a little bit more reliable. These are faster, obviously because they're rapid, slightly less reliable. And when I talk about how reliable, how many false negatives. So you're a little bit more likely to have a false negative with this than this, but that's improving all the time. And
Yeah.
last week in the faculty update, I think the president said that we're hoping to get rapid tests by the end of the semester, which would be great. Because you don't have to do so much quarantining and waiting in that case.
So that means there's gonna be a second semester on campus?
So far, I would say yes.
Oh, okay.
Are you
I mean,
sad?
oh my gosh, I get to come back.
You haven't been academically quarantined yet, I can tell.
No, I mean, I've been at home for 2 months, like.
It is not as good to have to join class remotely. So by the time you quarantine people when they're already having symptoms, they could have been spreading it. They could have been in this state, 2 days earlier, spreading it to other people. So the reason that King and other places are doing these quarantine rules is to stop the spread while it's still feasible to do so. And from my perspective, that seems to be working here at King. All right, who should get tested? Who in here has not been tested?
Like randomly tested?
Ever at all? That's really interesting to me. If you can't breathe well, you should get tested. That's just like a baseline rule. And if you can't smell or taste suddenly, you should get tested. And then if you have combo symptoms, like you have a fever and a cough, or if you have a fever and a headache, then you should. And I want to add one other onto here for you all. If you've been in a car with somebody who tested positive, like within that time period, I don't care how short of a time it was. I had this happen to a student. Like, if you're in a car with somebody and you've got the windows up, whether you have on masks or not, if you're in the car with them riding and they test positive, you need a test. Because that's small air recirculating and that's dangerous. And King won't tell you that, but I will tell you that.
So
Be careful. If you're going to go in a car with people, roll the windows down while you still can, and wear your masks. That's really important. That's just my advice to you.
So a close contact is considered somebody within 6 feet of an infected person for at least 15 minutes.
Yeah.
Is that with or without mask?
If it's not both in surgical masks, it doesn't count.
Okay, so
So
that would just be-
that's why we're all wearing surgical in labs, so that if you and I, it's basically would just be you and me.
Yeah.
If I talk to you for 15 minutes,
We would,
we
if one of
wouldn't
us
have to
tested
quarantine for
positive,
each other.
then,
Yeah.
okay.
And this happened to Professor Rudd. You know how he is. He's
Yeah.
a worrier. He went and got tested. The masks work. It's gonna be okay. So, but that's why we're
I just
doing
wasn't
what
sure
we're
like
doing.
if it was considered with or without mask. Or
No, I
something.
remember when we first heard that, the 6 feet, 15 minutes cloth masks won't cut it. Dr. Vaughn was like, okay, we're getting surgical masks for everybody in lab because we're going to be quarantining all over the place if that was the rule. So it's gone well so far. Gone well. This is kind of about the types of tests. I'm kind of going to skip past that. Okay. If you have friends or people that you know that tell you that this is the same as the flu and it's not worse than the flu, it's a lot worse than the flu. It's a lot. So in a bad flu year, like this 2018 winter, 80,000 people in the US died that whole calendar year of flu. 80,000 in 12 months. And in the US, we've really had this going for March was when we started testing for it. And we have 201,000 deaths in about 6 months. This is not the same as the flu. There's some advantages. Kids and babies and younger people are less hit by COVID disease than they are by flu. Like, can you imagine if kids were as susceptible as older people? It would be total panic. And when I went to talk to the 5th graders, I told them, like, this is not going to affect you, but your teachers are more concerned about it. Fortunately, most of their teachers are kind of young-ish at their school. This is a week old for Tennessee. We're right here. Okay. So we're right here, but this is a little bit misleading because we have a large population in this county too. So this, Bristol's like right here and Kingsport is over here. This is where Johnson City is. So we have a lot of cases, but we also have a lot of people because this is not per population. Our county has 30, no, let's see, maybe 44 deaths or something like that. This is a week old, but most of the people testing positive are youngish. Youngish.
I'm not 21, so it doesn't.
Y'all are over here. Probably in this class. Y'all are over here.
Not yet.
Okay, so
Ew.
this is my fifth grade leftover picture. Obviously, if you cough or sneeze out the virus, that can spread it. That's kind of obvious.
That's why we're wearing the mask.
This is the not obvious. So the first cold day, I like to go out and do dragon breath because it's special. We do that all the time. You can just only see it when it's cold. And this to me is the most important picture that I can show anybody related to anything coronavirus, is that we breathe like that all the time. So 3 people in a car with the windows up, mask or not, you're like in a fog of breath in there.
I guess I never realized that.
I mean, I know it, but I don't think about it until now. And like knowing that changes how you see the virus. So outside, it whooshes away. All this will disperse in the wind. But if you're inside and there's no air movement, that's like the scariest thing in the world to me right now. To go in a stuffy place.
Mm-hmm.
If you go in a stuffy place, just get out if you can.
I'm attending on that.
Where's your stuffy place?
Do I?
Do you have a stuffy place?
Oh, no.
Yeah, so this is actually why we do masks. So masks will block that going out if they're the right kind of mask. Okay. This slide, we'll come back and talk about this when we talk about disease transmission lecture, but here's a person breathing out drops. So this is like dragon breath, boy. We breathe virus out in drops. No, we don't breathe out any virus that's alone. Every virus that comes out of our breath or our sneeze is coming out in a droplet. That's a thing that the general public doesn't understand at all, and why would they? Why would they? Viruses come out in moisture. So all the virus we're breathing out is in some kind of droplet. If the droplet is big, it's going to fall down before it goes far. And this, let's see, drawing that to the wrong place. Okay, so the 6 feet rule, that's putting us outside of the large droplet range because they're going to fall down. And a lot of them are going to get caught in our mask too. But the small droplets are lighter and they can kind of disperse. They can stay in the air longer. And so if you hear about aerosols or the virus being airborne and people arguing about whether it is or is not, that's the far floating tiny droplets. This is me telling you my professional knowledge base. Most SARS transmission is this, but this happens some. It's mostly this. This happens some. But masks help catch that. Okay, this is a little bit hard for you all to see, but I can't find anybody that has made this picture except for my own bad drawing self. And this will be where we stop for today. Okay, so here's the surface of SARS virus with spikes. That's CO2. That's oxygen. That's how big they are. So if you've heard people say that masks trap CO2, they don't. CO2 is so small, it goes through the fabric just fine. So does oxygen come through the fabric just fine. The virus is a lot bigger, and the virus, remember, it's traveling in droplets. So imagine, the droplet is this big that the virus is traveling in. Oxygen and CO2 are not even in the same ballpark size of that. This is also why you can smell through a mask. Molecules that are small and aerosolized and have a smell, they'll go through a mask because they're a gas. They're just tiny. So
for like those people who say like, oh, I can't breathe through a mask, tell them they're full of.
No, that's real because I've felt it.
Mm-hmm.
Do you remember when you first started wearing them and you felt like, oh my God, I'm going to suffocate? Or maybe this doesn't happen to you, but the first time when I had to climb up 2 flights of stairs in the mask, like it's almost like a panic attack. Like I am going to suffocate, and I've had to get used to it. So when people first start wearing it, I know they feel like that because I felt like that. Mm-hmm. But it's still important to wear it. Do you all have a class right after?
Yeah, practice.
Your practice? If I could take 5 minutes and give the exam review, I think that would be helpful.
Oh, yeah.
Or just the exam list. The exam list, that would be really helpful. Okay, exam, what in the world? Exam topics. Our exam should be Friday, right?
Yes.
Yes. Exam will be Friday. Okay, so the first topic is general characteristics of enzymes. And generally, what's the difference between anabolism versus catabolism? Okay, that's big topic number one. Big topic number two is compare, contrast aerobic respiration, anaerobic respiration, fermentation, as in what do they require, how many ATP are made, where is their location, and what are their important waste products? What's the waste product of aerobic respiration? CO2 and water. Normally I used to joke about people like, I'm blowing it on you right now. CO2 and water. This one would be the wastes of anaerobic are CO2 and then some other thing. You don't have to know the wastes specifically of this, but what are the fermentation wastes that we care about?
CO2, acid, alcohol.
Yeah, CO2, acid, alcohol.
I'm learning.
You are. I'm so glad. Okay, so we have those things. So kind of the concept of DNA produces RNA, produces protein. I kind of had to go a little bit too fast through that. So basics of that. Then we talked about viral components. So remember, there are 5 viral components. There are 5 viral life cycle steps, 5 of those. The life cycle steps, I think I have you list them, and that becomes your word bank, and then you answer questions about them. Lytic versus lysogenic lifestyles. For influenza, the major thing there is antigenic drift versus shift. Antigenic drift versus shift. I really don't know what I'm going to put on for SARS-CoV-2. And then from Wednesday's lecture, compare, contrast, and know details of conjugation, transformation, translation. I haven't covered that yet, but that will be Wednesday.
And then is the test going to be all multiple choice?
No, never.
Besides, like you said, the one free response where you just list the 5 virus life cycle.
Everything that I wrote down as major topics is less likely to be multiple choice. So there will be multiple choice, and some of these things may be in some of the multiple choice, but it's kind of, it'll have a similar format to the last exam, except I think that there's less drawing on this one.
Okay.
Is there anything we have to draw?
Or any math.
There is no math on this beyond knowing numbers of ATP, really. Oh, we're not even that late. Okay, that's worth doing. I better take a picture of it too. There is a quiz on Wednesday about DNA recombination, which is translation, transduction, and conjugation.
Hey, how can you see the board?
So can you still send the notes? I think I missed some stuff.
Okay. Bless you. You're welcome.
I'm glad you have on a mask. Joseph Ong, our youngest son, like he'll come home and his mask has like a total wet spot right here. Gross, because he totally just sneezes, completely sneezes. But it's not really spreading in their school, so I'm so glad.
I really think I